Hi, my name is Sandy. I'm a rising junior in the mold department, and this summer I work with the University of Tennessee Health Science Center in collaboration with the Bonner Children's Hospital under my supervisor, Dr. Joan Hong. So me and my partner, Chino Eke, primarily worked in two labs, Dr. Han's pediatric obesity lab and Dr. Jack Sal's neurological lab, which focuses on amputation and phantom limb trauma. And so within Dr. Han's lab, we primarily focused on two medications, loxapine and atomoxetine, as potential treatments for obesity. These two drugs are typically used for psychological conditions. Loxapine treats schizophrenia and atomoxetine being used for ADHD. But they've had shown side effects in the past of weight loss or metabolism change. So we wanted to look into that and see if it could have effect on the mice in the lab. And so specifically, we wanted to see if these drugs would affect patients with mutations in the MC4R gene. The MC4R gene has been implicated in obesity in the past. And so we specifically performed this experiment on mice who had their MC4R gene knocked out. At this point in time, the experiment has already been done. And so me and Chino's job was mainly to analyze the data that had been collected through the CLAMS, which is a comprehensive lab animal monitoring system, in R, and to create graphics and to do statistical analysis in order to see if the drug had significantly changed activity, metabolism, or food intake in the mice. So excitingly, we actually found that it did decrease metabolism and activity as well as food intake in mice with the MC4 knockout gene during the drug phase of the experiment. And so we're really excited about these results, and we will see if in the future this will allow for a more expanded study or if we can publish a paper about this. But yeah, so we would still have to do some further research and analysis, but these results have seemed really promising. In Dr. Sal's lab, we mainly worked with mRNA expression data of the fly brain collected after fly leg amputation. We were interested in the potential differences in gene expression between like a rapid response after amputation, about 1 hour after it happened, and a delayed response about 5 days after amputation. So we focused on genes that were mainly up or down regulated by 1.5 times, and we investigated their function and potential human homologs and orthologs in order to see if we could use this data to somehow have some sort of clinical application or human application in the future. Using a database called DAVID, we were able to group them and functionally annotate them, and we interestingly found that there was a really large neurological immune response to periphery limb damage. So far, this is the primary large finding that we have, but we'd like to do some more research and also do a more in-depth literature search to see if something similar has been found in other organisms. And if that's the case, we would like to potentially do a study with mice or some other model organism in the future to see if it's applicable to humans. Outside of the FLY project, we also worked on drafting a paper, also regarding phantom limb pain, but in humans, in order to investigate the difference in gene expression between patients, amputee patients who have phantom limb pain and who do not. If we, as we continue to work on this paper, we're hoping to publish in the American Journal of Human Genetics. And yeah, so it's been a really, really fulfilling summer. I've really enjoyed this internship. It's been really great working with people, even though everything has been virtual. It's been great seeing the more back-end side of the research. So previously I had mainly worked in the lab, collecting the initial data, but being able to see that further analysis and even the drafting of the future paper has been really, really cool. And it's really opened up my eyes to what research can be like. I'm really, really thankful for everyone who I worked with, Dr. Han, Dr. Sal, the members of their labs, such as Dr. Kinney or Dr. Pyweller, to name a few, who were really helpful in giving guidance and who were always open to speak and to answer any questions that I might have. In general, this internship just confirmed my interest in medicine. It confirmed my interest in doing clinical research in the future. And yeah, it was just really exciting being able to do research that I knew could potentially help patients in the future. Yeah, so thank you so much just to everyone who has made this opportunity possible. Thank you to the Global Health Center. And yeah, thank you so much for listening to my presentation.